Okay. All right. Well, hello, everyone. I'm Sarah Smith, and it's my pleasure today to introduce our seminar speaker. But before I do that, I'm going to go over a few brief Zoom instructions. All audience participants will be muted throughout the seminar. Just a reminder, don't attempt to turn on your video or share your screen during the talk. And there will be an opportunity for the audience to ask questions live at the end of the talk, just like a normal seminar that's in person. Once the speaker has finished their talk, you can use the Zoom's raise hand feature. It's located under the reactions tab. We all probably know where that is now. You're invited to turn on your video during the questions portion of the talk. All right, so with that, I will move on to introduce our speaker today, Dr. Alyssa Griffin, who's an assistant professor of the Earth and Planetary Sciences and a PI of the Coastal Health and Nearshore Geochemistry Lab, or the Change Lab, which is such a great name for a lab, at UC Davis and Bodega Marine Laboratory. She received her bachelor's and master's degrees in geology from Temple University and her PhD in oceanography from Scripps Institution of Oceanography down at UC San Diego. She's interested generally in the cycling of carbon and related elements in the coastal marine ecosystems. I was also happy to see, and it was really cool to learn how interested Dr. Griffin is in using science to build climate resiliency in under-resourced coastal communities, and your commitment to diversity, equity, justness, and inclusion as we look forward into the future for the next generation of geoscientists is really notable and impressive. In this area, she's received multiple honors, including a UC San Diego Inclusive Excellence Award, an induction into the Boucher Honor Society, and UC Davis Chancellor's Postdoctoral Fellowship, which is all really cool. We're excited to have you here at Moss Landing and to hear more about your research. So with that, I will just welcome you to our seminar series and turn over the microphone to you to start your seminar.
Thank you. Great. Well, first, I just want to say thank you so much for the invitation to speak with you all today, and my sincerest apologies that it didn't work out for me to be there in person, but I'm just up the road a ways, so hopefully we can either connect up at Bodega or down in Moss Landing another time. So today I'm going to essentially be giving you an overview of my research program that's at the nexus of marine sediments and the changing climate. So first, I thought, since we didn't get to meet in person today, that I'd share a little bit about myself and my research interests. So I'm a geologist and a chemical oceanographer by training, and generally speaking, I am most interested, as Sarah mentioned, in the cycling of carbon and other related elements in coastal marine environments. And this interest is generally referred to as marine biogeochemistry. And just as the name suggests, biogeochemical processes and cycles are influenced to varying degrees by biological, geological, and chemical interactions, which occur on really broad spatiotemporal scales. And because the oceans are such a significant component of our planet and the Earth system, marine biogeochemistry plays a really important role in local as well as global biogeochemical cycling. And understanding marine biogeochemical processes across all of these scales is becoming increasingly important as human-induced alterations to our planet continue to accelerate. And the ocean continues to be perturbated by anthropogenic pollution, including nutrient and sediment inputs, and of course, carbon. So I'm sure many of you recognize this graph, which is the famous Keeling curve, and it shows increasing atmospheric CO2 on the y-axis since measurements first began in 1958 along the x-axis. And the first time I even thought about the climate or studying the climate was around this time. I was actually a music major, studying to become a vocal performance major. who had to take a science elective and ended up taking a geology course because honestly, I heard it was easy. But I was immediately humbled in this course by just the sheer size and scale of planetary processes, as well as geologic time, but also really fascinated that humans, as relatively insignificant as we are, could cause global scale changes in the Earth system. So this was way back pre 400 ppm. Currently, atmospheric CO2 is well beyond the threshold that we sort of referred to at that time as a tipping point. I pulled these numbers off of the Keeling Curve website this morning. So today's reading was 421.96 ppm, and we're well over 400 ppm throughout the year now. And because we've reached that threshold, we now know that in order to avoid complete climate catastrophe, we not only need to aggressively curb emissions, but we actually eventually need to reach negative global emissions. So we need to be pulling carbon dioxide from the atmosphere. And I really believe that climate change is the most pressing environmental and social issue of our time. And my research continues to be driven and motivated by a desire to better understand our climate system as a whole, our role within it, assess present climatic changes as they're happening, and also mitigate future climate consequences. So why do I study marine sediments? Well, marine sediments are the ultimate sink of atmospheric carbon dioxide over geologic timescales. So marine sedimentation really is a fundamental control of ocean chemistry and atmospheric carbon dioxide, and in turn, Earth's climate. And additionally, much of our climatic record, especially through deep time, is contained in ancient marine sediments and the rock record, which help us unlock past climates in order to help us better prepare and predict for the future. And I see biogeochemistry of marine sediments as a tool to not just understand individual processes, but to really build connections across these really broad time and space scales that we're talking about. So for marine sediments, these processes include those happening on the micro and very short time scale, such as mineral, water, and organic interactions, biomineralization and microbial activity, up to larger scale processes such as community and ecosystem dynamics, and sedimentary budgets. And all of these things are influenced by sedimentary biogeochemistry. And this influence even is seen at the global scale and over tens of thousands of years, and has importance in understanding climate change, as well as large-scale processes like ocean acidification, which influence and are influenced by marine sedimentation. So I would argue that in marine sedimentary biogeochemistry, we have a fairly good understanding of these individual processes at these broad scales. But I think we really need to improve our ability to build connections between them across time and space. And so my research really centers around using marine biogeochemical research to build these connections through space and through time. But most importantly, because our science doesn't happen in a vacuum, to build connections between these processes and between people. And so those connections have driven me to seek out and try to answer some of these broad research questions, which I'll read to you. So what controls early diagenetic processes in marine carbonate sediments? How will changing ocean conditions impact shallow marine carbonate budgets? And is carbon storage in marine sediments a viable climate mitigation strategy? And so, like I said, I really wanted to provide just a broad overview. So I'm going to address these questions through 4 themes that I feel sort of encapsulate my research. So first, I'll discuss how we can learn more about early diagenesis in modern carbonate sediments and how diagenesis influences our interpretation of the marine carbonate record. Second, I'll discuss the role of geochemical dissolution in shallow marine carbonate budgets. And then I'll address some of the outstanding questions in carbon cycling and storage in coastal marine sediments, as well as their potential application to proposed climate mitigation strategies. And then lastly, I want to share something that's deeply interwoven with all of the work I do, which is how these research themes and others can support and serve community-led partnerships that build coastal climate resiliency. So I have these icons on the top right of the slides to help guide you through the talk. And with that, we'll jump right in. So in order to discuss diagenesis in modern carbonate sediments, we first have to talk about biomineralization. So biomineralization is the formation of minerals by any organism, and biomineralization can heavily influence the geochemistry of marine tests, shells, skeletons, and in turn, our interpretation of these minerals in the rock record. So biominerals are very diverse and can be silica-based, like these diatoms and radiolaria. It can be phosphate-based, such as fish bones and teeth, or carbonate-based, such as nacre or mother of pearl, which constitutes pearls and abalone shells. But the most widespread and abundant biogenic mineral is calcium carbonate. And all of the marine organisms that I've pictured here precipitate forms of calcium carbonate as their structural and protective hard parts. Everything from these microscopic plankton to various types of shellfish that are critical protein sources for cultures around the world, all the way up to foundational species such as corals that support some of the most biodiverse ecosystems on the planet. And there are multiple abiotic and biogenic carbonate mineral phases in the ocean, including calcite, aragonite, and magnesium calcite. And they all have similar or the same chemical compositions, but they have different properties that influence their geochemical behavior. So these are just some SEM images just to show you how distinct these different crystal morphologies can be. So these are 4 carbonate polymorphs from left to right. We have calcite, aragonite, amorphous calcium carbonate, and the much less common vaterite. So like most minerals, biogenic carbonates can have significant physical and chemical differences from their abiotic polymorphs. So the figure on the left shows images of biogenic calcite on the top and abiotic calcite on the bottom. And all I want you to notice is that the surface architecture and structure of the biogenic minerals is much more complex and rough and textured compared to the relatively smoother surfaces of the abiotic grains below. And these rough surfaces lead to higher surface energy, which increases adsorption. And additionally, most biogenic minerals also have a organic matrix that's associated with them that makes the mineral less crystalline. And what this does is it actually leads to greater elemental substitution and higher incorporation rates in biogenic minerals. And of all these differences, and all of these differences interact and influence how biogenic minerals behave and interact with surrounding seawater, as well as their solid state geochemistry. So biogenic carbonates can accumulate in many marine environments, but some budgets suggest that nearly 50% of all marine carbonates accumulate in shallow marine environments. And these shallow carbonates may lithify and represent at least 70% of carbonates that end up preserved in the rock record. So why am I telling you this? Well, This means that many of the inferences that we make about changes in global climate and seawater chemistry over geologic time, they rely on interpretations of geochemical signatures contained in shallow carbonates. However, these interpretations often require the very important assumption that major physical and chemical changes haven't occurred after the mineral is deposited, which for the non-geologists in the room is a process known as diagenesis. And this assumption is much more appropriate in deeper sediments where we don't have any light and there's little, energy. But we know that in shallow sediments, photosynthesis and respiration, which take up and release CO2 respectively, occur in the sediments over daily cycles. And these daily changes in sedimentary CO2 levels are responsible for driving daily cycles in carbonate precipitation and dissolution as well. So typically you have photosynthesis happening during the day, drawing down CO2 and precipitating calcium carbonate. And at night you have the reverse where you're releasing CO2 and dissolving those carbonate minerals. And there are two parameters that we typically measure in seawater that sort of indirectly tell us whether the processes I just described are occurring. So the first is dissolved inorganic carbon, which just as it sounds, tells us how much inorganic carbon there is dissolved in seawater. And the second is a little trickier. It's called total alkalinity, which is seawater's ability to neutralize acid. And so total alkalinity is generally related to the amount of negatively charged ions in seawater, and I've listed the primary ones here in this equation. So changes in dissolved inorganic carbon and total alkalinity due to photosynthesis and respiration, as well as carbonate precipitation and dissolution, can lead to really strong chemical gradients between the sediments and the overlying seawater. And this results in fluxes of both dissolved inorganic carbon and total alkalinity across the sediment water interface. And by measuring these fluxes across the interface, we can actually learn more about these biogeochemical processes that are occurring within the sediments. So this is the approach that I would say is most widely used by researchers in my field. But unfortunately, just measuring dissolved inorganic carbon and total alkalinity is actually somewhat limited. So for example, this method can't differentiate between the dissolution of calcite, aragonite, or magnesium calcite, because dissolution of any of those minerals increases total alkalinity by the same amount. And additionally, there's all these other processes that can influence both DIC and alkalinity, such as redox dynamics and groundwater inputs, as well as organic acid-base systems. And so because of these interfering processes, sometimes the carbonate dissolution and precipitation dynamics can be somewhat difficult to parse apart. And so these limitations make it really difficult to answer some critical outstanding questions in the field, which include, we know things are precipitating and dissolving, but which of the carbonate mineral phases are participating in those reactions? And then also what controls the apparent solubility, which is just the likelihood of something to dissolve, because we've seen things like mineralogy, microarchitecture, grain size, all kinds of things play a role in how likely something is to dissolve. So using a wide range of both laboratory and field-based experimental approaches and additional geochemical tools, I've tried to find answers to these unresolved questions. So I'm just going to talk about one tool that I've used to try to get at this question of what mineral phase is dissolving. And that's using trace elements associated with carbonates. So due to their differing crystal structures, different carbonates incorporate different elements such as strontium and magnesium preferentially into their structures. So magnesium tends to be incorporated preferentially into calcites and strontium into aragonite. And they substitute for calcium, which is the green atom shown here on the right in the crystal lattice diagrams. So theoretically, by measuring differences in strontium to magnesium to calcium ratios, we should theoretically be able to determine which carbonate phases are preferentially dissolving and participating in these reactions. So for the sake of time, I'm not going to discuss this work in much more detail today, but using this approach, I was able to demonstrate that using coupled calcium and alkalinity measurements can actually improve our estimates of carbonate precipitation and dissolution dynamics, even at the reef scale. So our study site was the Bermuda Carbonate Platform. And if you're interested in learning more about this work, it was recently published in Marine Chemistry, and I invite you to check it out. So I also just want to quickly note that these techniques and these types of questions are, again, really important to understanding geochemical proxies and how we use them to interpret past climate and past ocean conditions. So as I mentioned, diagenesis is such a problem when we try to go into the rock record and learn more about what environment these rocks were formed in. And a recent review paper came out that shows how different geochemical proxies, how susceptible they are to diagenetic alteration. And you can see here under mineralogy that magnesium, strontium are used for things like temperature and ice volume, mineralogy, but they're also, this big red arrow shows that they're extremely susceptible to diagenesis. So how can we work around this and better understand diagenesis so we can improve the use of these tools to interpret past climate? Well, we can not only measure the fluxes of DIC and alkalinity, but we can also measure these metal ratios both in the sediments and across the sediment water interface. And understanding those processes and those fluxes can help us correct for diagenetic signals that we see in the rock record. So some of my upcoming work is using highly precise measurements of these metal ratios using ICP-OES in both pore waters and overlying waters on carbonate platforms to really refine our understanding of these early diagenetic alterations. So moving on to our second question, what controls the apparent solubility of shallow carbonate minerals? So for abiotic or non-biogenic minerals, we tend to think of solubility simply as a function of mineral stability or thermodynamically. However, in biogenic substrates, it's been shown that mineral solubility can actually be overridden by these other properties that I mentioned before, such as grain size, surface architecture, and organic mineral interactions, all kinds of different properties. And it makes sense that shallow carbonates would behave very differently from, say, abiotic calcite like this Icelandic spar, or more homogeneous deep sea sediment like these coccoliths. Because as you can see in this picture of shallow carbonates on the bottom, these biogenic sediments are highly complex and very heterogeneous, and they come from a variety of sources. So we see this complexity reflected even at the micro scale. And I wanted to show you just how different these grains can be. So these are two grains from the same subsample of sediment. And you can see on the left, I have a little benthic foram. And on the right, I have this little cute gastropod. And these are tiny grains within the same sample. And this heterogeneity at the very small scale makes it really difficult to attribute larger scale processes to any of these microscopic characteristics in natural sediments. But despite this difficulty, it's widely accepted that carbonates are expected to follow certain paradigms of carbonate dissolution, which include that dissolution should increase with increasing PCO2 or decreasing pH, decreasing grain size because of an increase in surface area to volume ratio, and decreasing mineral stability. But I wanted to explicitly test these paradigms specifically in shallow carbonate sediments. So to do that, I measured the dissolution rates of well-characterized carbonate sediments from both the Bermuda carbonate platform as well as the Heron Island platform in Australia in a controlled laboratory setting using the reactor that I pictured here. For time, I'm not going to describe the reactor, but if you're interested, I'm happy to talk about it more. So this work I do want to mention was done with the help of 3 students who were undergraduates at the time. They're now PhD students, Morgan Goodrich, Sam Kikueva, pictured here on the top, and Ralph Torres also pictured. And the results of these experiments are pictured here in the graph. And as anticipated, the dissolution rates shown here on the y-axis of all of these various samples increased with increasing PCO2 on the x-axis. What was interesting, though, is that I found no correlation between the measured dissolution rates and the grain size or the mineralogy of the sediment samples. In fact, of all the physical and chemical characteristics that I analyzed in these samples, the only significant correlations with increasing dissolution rates that I was able to identify were with decreasing organic content and decreasing delta 13C of that organic content. So where are these patterns coming from? One possible explanation for these trends could be related to organic mineral interactions. So like many biogenic minerals, your bones are a great example. Biogenic carbonates have an organic template that's tightly bound to the crystal structure. And this organic matter is known as intracrystalline organic matter. In addition, it's been shown that organic molecules can absorb or attach to the surfaces of biogenic carbonates, which is referred to as non-intracrystalline organic matter, which I demonstrated with the animation on your right. And it's been previously suggested that these organic coatings may actually act as a sort of protective barrier around sediment grains and therefore inhibit the direct interaction of seawater with the mineral surface. And obviously, that's going to influence your reaction rates. But I wanted to test this. So in order to test the influence of these coatings on dissolution, I soaked the sediment in bleach, which removes only the absorbed organic matter without altering the mineral surface or the intracrystalline organics. And sure enough, when I measured dissolution rates of the bleached versus the unbleached samples of the same sediments, the bleach samples where the coatings were removed, which are shown in the dashed lines and open symbols here on this graph, had dissolution rates that were approximately 2 to 3 times higher for sediment samples relative to sediment samples that hadn't been bleached for both high organic content and low organic content samples. So these experiments could start to explain the trend that we see in total organic carbon and dissolution rates. But what about that isotopic trend where the dissolution rates increased when the delta 13C of the organic carbon also, decreased or became lighter? So to get at this question, I actually determined the isotopic signatures of the intracrystalline versus the non-intracrystalline organic matter. And once I did that, this trend also started to make sense. So I found that the adsorbed or non-intracrystalline organic matter has a significantly heavier signature of negative 11.8 per mil relative to the intracrystalline matrix, which had a signature of negative 13.8 per mil. So what this suggests is that as non-intracrystalline organic matter decreases on the grain surface, the overall signature would become lighter and more similar to the intracrystalline organic matrix. So if we piece all of this together, I know that was a lot. So if we piece all of it together, we can start to explain both trends in the organic matter data. So I want you to keep your eye on the animations on this slide. So I want you to imagine that as organic matter is removed from the surface of our carbonate grain, the total percent of organic carbon will start to decrease, which is shown here in orange on the left axis. And as that heavier non-intracrystalline organic carbon is removed, the overall isotopic signature also becomes lighter, which is on the purple axis here on the right. But now, because that absorbed organic matter is removed, more of the mineral surface is exposed to surrounding seawater, which increases the dissolution rates shown here on the x-axis. So all of these findings really support the hypothesis that biogenic carbonates, their dissolution rates decrease in the presence of these organic matter coatings on the surface of grains, again, because the mineral surfaces are unable to directly interact with the surrounding seawater. And this might not seem important if you don't think about these things every day like I do, but this really challenges some long-held assumptions about carbonate dissolution, and in turn, the cycling and geochemistry of carbonates in marine environments, which of course is linked to the carbon cycle and our climate. So the next theme I'll be discussing is dissolution and shallow marine carbonate budgets, and in particular, carbonate budgets in coral reef systems. So typically, reef growth is measured using a term called net community calcification, or NCC, which is just the balance of gross calcification and gross dissolution. However, I want to argue that net community calcification is really a predominantly biological / chemical view of reef systems. If we look at reefs through a geologic perspective, we can now incorporate processes other than just biological calcification and geochemical dissolution that impact a reef's ability to accumulate carbon. So these processes include both inputs such as physical import and inorganic precipitation, and outputs such as physical export off the reef and bioerosion. So unfortunately, both NCC, net community calcification, and net carbonate accumulation on reefs are expected to decline with warming oceans and increasing acidity due to climate change, because all of the inputs on the left-hand side are anticipated to decrease, while all of the outputs are expected to increase. And what this means is that we could actually shift reefs into a state of net erosion or reef loss, or it could simply lower accumulation rates enough that reefs will no longer be able to keep pace with sea level rise. Either way, understanding the balance of these inputs and outputs and how they will change is really critical to predicting the future of reef systems. And of all of these processes, I'd say that organismal calcification's response to a changing ocean is by far the best studied. However, multiple studies have suggested that abiotic processes, such as dissolution, may be far more sensitive to future ocean change. And so I wanted to better understand the role of carbonate sediments to overall reefal carbonate budgets. And so I had a great opportunity to conduct experiments near Lizard Island, Australia, located along the Great Barrier Reef. If you get a chance to go, highly recommend. So I mentioned earlier in shallow carbonates, we see these fluxes of dissolved inorganic carbon and total alkalinity across the sediment-water interface. And that by measuring these fluxes, we can start to parse apart the role of various sedimentary biogeochemical processes and how they're influenced by changes in the overlying water column. So one method we use to measure these changes in sedimentary fluxes of dissolved inorganic carbon and total alkalinity is by performing in situ sediment incubations. So I've included this short video, that's me, sampling a set of chamber incubations from ports that they have on the lids using syringes. We can also use these ports to inject different treatments that mimic changes that we want to understand in the overlying water column, such as those predicted under future climate scenarios. So once these syringes are collected, we can take them to the surface to be analyzed for dissolved inorganic carbon, total alkalinity, and other relevant parameters. And the chambers extend about 20 centimeters into the sediments. And again, we infer the rates of the biogeochemical processes within the sediments by measuring what fluxes into the overlying chamber water. So these experiments were conducted at 4 contrasting sites pictured here along the southern shore of Lizard Island. So we measured net community calcification as well as net community production, or NCP, which is just the balance of photosynthesis and respiration. And those measurements were used to infer, or excuse me, those rates were inferred from changes in dissolved inorganic carbon and total alkalinity over time. So each of these incubations lasted 24 hours, and they were sampled at dusk, dawn, and dusk to calculate both daytime and nighttime rates, as well as the integrated rates over 24 hours. And we wanted to achieve a wide range of starting conditions in the overlying chamber water. So we introduced high alkalinity water to mimic past conditions in the ocean. And we also injected high PCO2 water to mimic future acidification scenarios. So before I show you these data, I just want to orient you to the figure. So NCP rates are on the left side, and NCC calcification rates are on the right. The data are grouped by site, which is shown along the x-axis, and replicates are bimmed by treatment, where acidified treatments are shown in red, control treatments are shown in green, and alkalinity enriched treatments are shown in purple. In the upper panels, nighttime rates are shown with this more transparent coloring, and daytime rates with the opaque coloring. And then the lower panels only show the integrated 24-hour rates. So if we look at the NCP rates across sites and treatments, we find no significant differences across treatments during the day, or at night. Which suggests that, so it just suggests that those processes are not influenced by changes in overlying ocean carbonate chemistry. However, the NCC rates show quite different patterns. So across all sites, we see that nighttime dissolution rates, again with the more transparent colors, were significantly different under the different treatments, with lower NCC rates under acidified conditions and higher rates under alkalinity enriched conditions relative to the present conditions shown in green. However, daytime NCC rates were higher under the alkalinity enriched treatments, but what was really interesting is that there was no significant differences in NCC rates between control and acidified treatments during the day. And this suggests that some mechanism is tempering the response of net community calcification rates to decreases in saturation state during the day, but those same mechanisms are not happening at night. And one possible explanation could be that during the day, biological calcification is the dominant process occurring, and it may be less sensitive to changes in saturation state because some organisms have been shown to control their chemical conditions near their calcifying surfaces. And this is really in stark contrast to gross dissolution, which is an inorganic process that's subject primarily, once again, to thermodynamic constraints. And so this difference that we see between biological calcification versus geochemical dissolution could explain why net community calcification rates decrease at night under acidification, but not during the day. And I think what's particularly important is that the integration of these rates over 24 hours results in significantly different net community calcification rates across treatments. And all of the acidified conditions at all sites transition from net calcifying, which is above that dashed black line, to net dissolving below under acidified conditions, again, at all 4 of the sites. And so what this tells us is that because there was no differences between acidified and controlled conditions during the day, it suggests that dissolution at night is primarily responsible for the transition from net calcification to net dissolving under acidified conditions. What it also suggests is that dissolution may be more sensitive to changes in ocean carbonate chemistry than biological calcification. So in the future, we may want to do more studies on carbonate dissolution and its response to changing ocean chemistry rather than organismal responses. Both are important. So the findings I've outlined here are important to predicting future changes in carbonate accumulation in shallow reef environments, which of course is critical to the survival of coral reefs. It also suggests that changes observed in net community calcification from previous in situ acidification and alkalinity enrichment experiments could primarily be driven by sedimentary processes and not by organismal ones. And I think these experiments and these findings are also really applicable as we move forward with climate mitigation strategies. So there have been several marine CDR approaches or carbon dioxide removal approaches proposed, one of which is alkalinity enhancement. So these experiments showed that when you alkalinize coral reef sediments, that you indeed are increasing calcification without releasing more dissolved inorganic carbon. So I'd like to look a little more to the future and discuss carbon cycling and storage in all coastal marine sediments and how it relates to some of these potential climate mitigation strategies you saw on the slide before. So coastal marine sediments are situated obviously within the coastal ocean, where carbon dynamics are a hot mess and governed by a multitude of drivers pictured here, which include impacts from watershed processes, ecosystem metabolism, and a whole suite of physical processes. And because coastal carbon dynamics are so complex, it can be really difficult to track the sources and sinks of carbon within the coastal ocean. And I think this difficulty also inhibits our ability to predict how individual processes, as well as the coastal ocean as a whole, will respond to future climatic changes. So predicting these changes has important implications, not only for local carbon dynamics, but also global carbon dynamics, because the coastal ocean really is an important link between the terrestrial and oceanic carbon cycles. So the exchange of carbon across the sediment water interface, shown here as sediment exchange, is really in desperate need of better understanding. So despite its potential significance at both the local and global scale, coastal sedimentary carbon exchange isn't well constrained, nor are the biogeochemical processes controlling them. And one of the reasons for this is because there are so many processes happening in these sediments that can influence carbonate chemistry in similar or sometimes even identical ways. So for example, this figure from a paper from me and my colleagues show changes in total alkalinity on the y-axis against changes in dissolved inorganic carbon along the x-axis. And the relationship between these variables drive the changes in pH that are shown by the color bar. And the arrows on the plot indicate how these parameters all change due to different biogeochemical processes. So we've talked about carbonate dissolution and precipitation, as well as respiration and photosynthesis a fair amount. But there are also all of these redox reactions, such as sulfate reduction and denitrification, that can also release alkalinity and dissolved inorganic carbon into the system. So the various biogeochemical processes occurring in the sediments now are even trickier to parse apart because we're now including all of these other interactions. What's interesting, though, is that recent work has suggested that lateral export of inorganic carbon from vegetated coastal ecosystems, such as mangroves, may be a significant oceanic carbon sink that we're not accounting for. And in fact, when we compare organic carbon storage in these sediments relative to this inorganic carbon lateral flux, it's been suggested that that flux could be up to 4 times larger than the traditional blue carbon sink of buried organic material. And that's true for all 4 major vegetated coastal ecosystems. However, most of this work has been done in mangroves, and seagrasses are severely understudied in this context. And I think that's unfortunate because seagrasses, represented on this distribution map by green dots, are actually the most expansive of the major vegetated coastal ecosystems, and are present along the coastlines of every continent except for Antarctica, including in my scientific backyard at Bodega Bay. And so what we've been doing recently is trying to characterize how sedimentary exchange, how sedimentary exchange is influencing coastal carbon dynamics and also these lateral fluxes of inorganic carbon to the open ocean. And some of our future work is trying to look at these, look at seagrasses that are growing in very different sedimentary environments. So there are the seagrasses that we have here in Northern California that grow in organic and iron-rich siliciclastic sediments versus tropical or subtropical seagrasses that can live in carbonate-dominated sediments. And we would anticipate that the biogeochemical processes that are producing or that are transforming carbon in those systems to be very different. And so right now we're using our access to seagrasses in siliciclastic muddy sediments right outside the lab to start constraining these fluxes of carbon, both inorganic carbon and organic carbon in the system. And so this is a picture of Manny Delgado, my laboratory technician, and Lupe Carrasco, an intern in our lab, collecting samples in Bodega Harbor. So stay tuned. I think some really interesting stuff is going to come out of that work. So I know I'm running we're getting close to time here, but I do want to make sure to get to this last section because, as I mentioned before, it's really inseparable from the work that I do. So I just want to take a little time to talk about these community-led partnerships and why I'm so passionate about participating in them. So unfortunately, there's often very little overlap between who's impacted by climate change, who's doing climate change research, and who's making policy decisions related to climate. And this is really driven by the fact that there's a lack of diversity among climate researchers and policymakers. And this lack of diversity is caused by exclusionary practices in the way climate research and policymaking is conducted. And these exclusionary practices exclude the very people who will be most impacted by climate change, which we know are Black and Indigenous peoples, communities of color, women, and low-income populations. So I want to highlight just two community-led partnerships that I've had the privilege of co-creating and participating in. And the first is through AGU's Thriving Earth Exchange. If you haven't heard about Thriving Earth Exchange, I encourage you to Google it. It's a way for you to get in contact as a scientist with communities that are reaching out for your help. So I was approached through the Thriving Earth Exchange to develop a risk assessment report for a proposed limestone mine near a coral reef in North Andros, Bahamas. And this project is led by a group of local fly fishermen and the Bahamas Agricultural and Marine Science Institute. And I'm really excited to say that some of our work has led to the dismissal of the mining proposal, but unfortunately, we know they have lots of tricks up their sleeves and will likely be back with a different proposal. So the fight there is still happening. The second partnership is with a local community-led environmental justice organization called Green Action. So this project set out to analyze and map legacy contaminants from the decommissioned Hunters Point Naval Shipyard in San Francisco Bay. And this work was in collaboration with Hunters Point Biomonitoring Program, which continues to identify and monitor disproportionate health issues and cancer clusters in the surrounding neighborhood of Bayview, which is predominantly a low-income community of color. And so these are just some of the ways I found to use my skills in marine geochemistry, as well as my experience in environmental consulting, to really center and support the goals of vulnerable and under-resourced coastal communities. As a researcher, I also strive to use my privilege to increase accessibility and inclusion within climate research. So for those that don't know, the geosciences are the least diverse among the STEM fields and have made very little progress towards diversifying in the last 40 years. So if you haven't read this paper by Bernard and Cooperdock, I really, really emphasize reading it. So just to give you some numbers, currently less than 5% of all geoscience degree recipients in the US are women of color, and less than 1% of all US geoscience faculty are women of color. But as a woman of color, I don't really need these statistics to tell me that. It's something that I and all of those who came before me have experienced and continue to experience firsthand. And I can tell you, when you're the first or the only in the room or both, it leads you to really question your belonging, which is not a great feeling. So I try really hard to not only hold the door open behind me, but really just take the door right off the hinges. And so these are just some of the programs that I've had the pleasure of developing, one of which is a scholarship through Scripps Oceanography to provide funding and support and mentorship for marginalized divers, scientific divers, so they can gain the training they need to use diving as a oceanographic research tool. And the second is starting this year and was funded through California Sea Grant to provide mentorship and community building for, most importantly, undergraduate research experiences for our incoming coastal and marine science transfer students at UC Davis. And, I really try to support programming like this because broader participation in climate science matters. There's a quote that I first heard from Dr. Brandon Jones at NSF, who often says in his talks, we're facing all hands on deck issues, but not all hands are on deck. And so I like to encourage everyone to see how they can build bridges both within and between your own personal and professional communities, because the only way we're going to tackle this problem and address the climate crisis is by doing it together. So that's all I have for you today. If you'd like to learn more about our research group or reach out to me and connect with us, again, the Coastal Health and Nearshore Geochemistry Lab, aka the Change Lab, you can visit us at ucdchangelab.com. And I always like to end my talks with a reminder of why I do the work that I do, which is my favorite geologist in the world, my daughter Ramani. And with that, I'll take any questions you may have. Thank you for your time.
All right. Can you hear me?
Yes, I can.
Excellent. Okay, so I'm going to be moderating any questions. Are there any questions in the room? I'll keep an eye on the chat as well. All right, we've got a question from Amanda Kahn. Okay.
Thank you so much for your talk. That was, I learned a lot. And coming at this from a biologist perspective, I'm not sure if I'm asking a question that actually is askable. So when you were showing the treatments where you were measuring net community productivity and calcification rates, NCP and NCC, I wondered whether it was possible to modify both CO2 concentration and alkalinity in the same containers, and whether, I don't know if those conflict each other, but if it did, do you think that there would be the same effects, or can they cancel each other out? Is it synergistic? Is it opposing? Yeah.
Yeah, no, that's a great question. I'm just gonna flip back to one of my plots real quick because I think it will be helpful. If you look at this plot here, you can see that in the background is change in pH, right? Which is oftentimes what we measure as a variable to determine biological response. But you can also see that the change in pH is really dependent on the ratio of change between total alkalinity and DIC. And what's interesting is that if you change total alkalinity and DIC in a 1-to-1 ratio, you actually don't change the pH very much. And so there's all these sort of levers that you can tweak that change the PCO2 and pH, don't change the PCO2 and pH, just change the CO2 content, just change the alkalinity content. And so, I think that answers your question, but if that didn't fully answer it, please let me know.
It definitely does. Thank you. That clarifies it too. I remember seeing this, but didn't understand it as well as I do right now. So thank you.
Awesome. Yeah, you're welcome.
All right. Are there any other questions in the room? I have a question, but I was.
I think
Nalina's?
I think I see one online. Yeah. Katie Duncan, maybe? I see a hand raised.
Oh, good. Yeah. Katie, go ahead and unmute yourself.
Okay, I'm unmuted. Can you hear me okay?
Yeah, can I hear you great?
So one thing I was wondering, you mentioned earlier that there was an initiative to manually decrease alkalinity around coral reefs or other coastal environments to encourage calcification. And then with your mentioning about like marginalized groups, especially like poor areas that might rely on these reefs, it seems like that would be, is it a costly thing to have widespread decreasing alkalinity, like an effort to decrease alkalinity? It just sounds like it would be very labor intensive and potentially costly.
Yeah,
alternatives to that.
Yeah, absolutely. So ocean alkalinity enhancement is something that's being proposed in a lot of different marine environments, not just in coral reefs. And this, marine carbon dioxide removal space is honestly just the wild, wild west right now. And I'm going to answer your question kind of with my opinion, which is that given the things that you're thinking about, that you're asking about, for those exact reasons, I think that we really need to be mindful about the impacts that these potential solutions will have on the communities that are most connected or most dependent upon these ecosystems. I don't think there's going to be a silver bullet. I don't think we can just go dump a bunch of stuff in the ocean and be like, poof, we pulled out a gigaton of carbon. I think it's going to have to be really hyper localized, and really be done thoughtfully and in collaboration with the communities that these proposed solutions might potentially impact. But that was a great question. In terms of feasibility, I don't think anyone's really thought that far yet. They're still trying to just figure out the chemistry.
Thank you. I was just thinking like who would pay for it, because it would be really bad if the community itself had to potentially pay for something that was costly. Anyways, thank you. Sorry about that.
No, yeah, so right now it's a lot of venture capitalism that's going on around the carbon capture market. So tech bros are paying for it right now.
Cool. So the one of the things I've heard of in this space is no silver bullet, but it'll be silver buckshot. So like all bunch of whole different tiny silver bullets thrown in the right direction.
I love that. I haven't heard that. That's perfect. Yeah.
All right. So we've got a question in the chat from Rachel. Rachel, do you want to unmute yourself and ask the question?
Sorry, I got like a notification. Anyway, yeah, no problem. So thank you so much for an amazing talk. That was fascinating. I am really interested in the redox dynamics that you were talking about and their influence on carbonate dissolution and precipitation, and specifically your how you might think like methanogenesis might influence those. That's
right.
particularly related to my thesis.
Awesome. Cool. I can see why you were hoping to chat today. Awesome. We'll find a time. Don't worry. So the redox reactions, it depends on which one. So sulfate reduction actually increases alkalinity. And so what can happen is that if you have enough sulfate reduction happening in these anoxic sediments, you can actually raise the saturation state so high that you're in situ autogenically precipitating calcium carbonate. And this is a little counterintuitive to a lot of folks, but when you precipitate calcium carbonate, you actually release CO2. And so that's something to think about as well. Methanogenesis is going to draw down your CO2, but depending on whether or not that methane stays buried is what you need to be more concerned about, because if it gets released in methane form, we know that it's a much more potent greenhouse gas. And also there's the potential for it to become, reoxidized. And so, there's a couple of different pathways where it can be problematic and challenging to understand what could happen. But generally speaking, methane's fine as long as it stays in the sediments, which is true of carbon too, I guess.
Awesome. Thank you so much.
Yeah, you're welcome.
Okay, so I've got a question for you too. The data that you showed on dissolution of sediments and showing that grain size isn't actually like all that important was really interesting in the context of the organic carbon that's adsorbed on the surface. Is that organic carbon from on intact shells, or is that stuff that adsorbs over microbial processing? And one question that came to mind is like, are there seasonal patterns then? And when you get lots of like storms that are breaking things up that would lead to dissolution versus in the summertime when there's calmer seas and you get more organic matter deposited on them?
Yeah, great question. All the things that keep me up at night. We don't know yet. So folks didn't even think that these coatings existed. And if they did exist, they didn't think that they were substantial enough or significant enough to influence chemical interactions. And so we're just starting to try and figure out, what is it? Where is it coming from? When does it form? All of those questions that you're thinking about. So one thing I'm really interested in pursuing is, using some omics strategies or omics approaches to look at different microbial communities and how they change in different sedimentary environments, and how what they're doing changes. I mean, I am not a microbial biologist, but these are some of the questions that we need to answer, because we know that all of these dynamics, even though they're influenced by overlying waters, are really driven by microbial activity in the microenvironments. And so the answer is we don't know, but we want to find out.
Cool. There are lots of experiments that you've got lined up then.
Yeah, well, I mean, my hypothesis, I think, is that they're adsorbed. And the reason I say that is because we actually see that these coatings are more significant on smaller grains. And that suggests it's a surface energy thing. So the bigger grains have less coating.
Cool.
So
it's very, very, very cool. Great talk. So we've got a question in the audience. This is Jacob.
Have that same question.
Oh, nice.
So I'm going to expand on that a little bit and just ask if you think that would apply to living organisms, like benthic organisms, that are essentially creating calcium carbonate surfaces in the environment.
Yeah, so there have been a few experiments done on this, but they've mostly been done on cultured organisms. And I personally have my doubts that cultured organisms are undergoing enough similar processes as they would in the natural environment to really develop these coatings. But with the cultured biogenic calcifiers, they did not see these organic coatings have an influence or even really form in those experiments. And so therefore they didn't really have any influence on their dissolution rates. But I imagine, if you have something living in the benthic environment right next to these grains that are getting this stuff absorbed onto them, I would guess that it's happening to the living organism as well. But how it manifests could be different.
Okay, great. Yeah, thank you. I'm just thinking about how the balance works with the accretion of that layer while there's dissolution going on, at the same time. So, and then how a living organism would mitigate that. But it sounds like a really cool area to explore.
Yeah, definitely. It's exciting stuff. I'd love to figure out ways to answer some of those questions because I think they're really important, but I'm biased.
Well, thank you so much for your wonderful presentation. So nice to host you here at Moss Landing, even virtually, and hope that you get down to visit us before too long.